Hello, in this video we will discuss about the biological nitrogen fixation. So the biological nitrogen fixation means the presence of the living organism will fix the nitrogen gaseous state into the amino acid and DNA repair, etc. So the formation of the living organism, because we are made up of nitrogen, carbon and oxygen and hydrogen. So let's begin to understand what is the nitrogen. So the nitrogen fixed through the action of microorganism is termed as biological nitrogen fixation because the living organism involving, and it can be aerobic, facultative and anaerobic. Aerobic nitrogen fixer is during the oxygenated environment, while the facultative will be deoxygenated or oxygenated environment. Both are favorable, while anaerobic is the without oxygen. So the free living bacteria and cyanobacteria is very important for free living and fix the nitrogen, while the symbiotic association through aerobic bacteria can be azotobacter during the night time, and the anaerobic bacteria is the photosynthetic can be, and the rhodosporidium and clostridium. This is the two type is the best example of this bacteria. So let's begin to understand the symbiotic fixation or symbiotic fixer bacteria. What is that and what that means? That is very important. A nitrogen fixing bacteria that occur in association with higher plants such as Leguminosae and Fabaceae family are termed as symbiosis and mutualism, and in this way the bacteria provide the nitrogen in the form of nitrites and nitrates and nitrites and nitrates, while in the case of bacteria will consume the energy through a plant and consume the other mineral and vitamin, which that is very helpful, and this helpful symbiosis is known as the mutualism because both are beneficial to each other. So here I am drawing the plant, which that is first of all which you need to understand the nitrogen cycle. So here is the plant contain roots. Roots contain nodules. After the development of nodules, we will discuss later in this video after the nitrogen cycle. So here the carbon dioxide will be absorbed and the water absorbed, as well as nitrogen will be assimilate through a soil by the bacteria, so the water will be absorbed to produce the energy and glucose formation. So let's begin to understand the nitrogen cycle. So after the dead animal will become a dead and decomposed. So after the decay, dead and decay matter will convert into the ammonia, that is NH3. And after the ammonia will be converted, and this ammonia by the fixation of the nitrifying bacteria will fix, and it fix into the nitrites NO2 negative, so the excess of the electron. And this nitrifying bacteria can be azotobacter and photosynthetic rhodosporidium and clostridium, which that is the independently, it is not symbiotic. So here is the I am drawing the atmosphere containing nitrogen. So the nitrogen is 79% in the atmosphere. During the thunder of cloud will producing the light, so the lightning will convert the nitrogen into the nitrates. So these nitrites and nitrates conversion, and due to the thundering, that is the important things you should need to understand, which that will be fixed by the bacteria. After the fixation of the bacteria through a, and also the denitrifying bacteria, which that is also convert the nitrates and nitrites into the nitrogen gas. So that is the bacteria also dissociate, and this dissociation of the nitrates convert into again nitrogen. So this is the nitrogen in atmosphere. So let's begin to understand the nitrogen in the atmosphere and what is the function. During the nitrogen is also plant and microbe mutualistic behavior in the nodules of the legume plant, which that basically fix the biotic nitrogen fix and this biotic nitrogen fixed in the form of nitrates and nitrites. After the assimilation into the plant, which that is convert the ammonia into the different type of amino acid, aspartate and glutamate. That is the major conversion to, and this plant will be contain nitrogen in the form of amino acid, protein, and this will be ingested by the animal. Digestion of the animal, after the digestion it will again dead, and the cycle will be continue for the circulation of the nitrogen throughout the earth. So here is the nitrogen fixer, can be free fixer or symbiotic. So the symbiotic fixer can produce the different type of protein in the different plants. For example, legumes plant which that produce a legume. So this is, you can see and understand, that is the beans and the grams contain protein due to the nodules are present which will fix the nitrogen in the form of ammonium ions. So we can't consume nitrogen through air, so we use food for consuming nitrogen in the form of beans. So that is why the nitrogen will not, when we will inhale the nitrogen, so the exhale equally not useful. So let's begin to understand the biochemistry. So the nitrogen cycle was the overview. You can learn more about the nitrogen cycle. If you like this video, please give the comment for the new video about the nitrogen cycle. So the biochemistry we will discuss, if first of all we should need to understand the nodule development. So the nodule development is the root excretes the substance that attracts the chemoattractant of the bacteria, which that is chemotactic movement of the bacteria toward the stimulate them toward the root is known as positive chemotactic movement, and stimulate them to divide. After the division of the rhizobia, so this rhizobia will be attract, and here is a xylem and cortex pericycle which that is dividable and thus basically produce substance, and this primary nodule meristem. Meristem mean the plant will be divide and differentiate the cell. So the bacteria attach themselves to root hairs. The the cell is in the pericycle near xylem are stimulated to divide and form infection threads. So the infection threads will be formed, and in this way the infection thread, you can see it is will be grow, the pericycle will grow, and nodule vascular tissue will be grow, and in this way the infected root hair formation will be occur, and in this way the bacteria are present in the near the nodule, and in this way you can see and understand in this way the cells of primary nodule meristem pericycle continues to divide, and two masses of dividing cells fuse into single clump. So the single clump formation will be occur, and in this way the nodule elongate, differentiate, make a connection with the root still infected, and the bacteria enter the nodule cells. So you can see the enter into the nodule cells. So after the entry into the nodule cells, so the nodule vascular tissue will be formed, and this nodule will absorb the bacteria, and bacteria will remain into the nodule for the fixation of the nitrogen in the form of ammonium ion. So the nitrogen will convert into the ammonia will convert into the ammonium ion, and that ammonium ion will be absorbed into the plant for the further transamination, as well as the amine formation, the amino acid formation. So the nodule elongate, differentiate, make a connection with the root still infected. The infected zone are present here is that you can see infected zone. So the bacteria enter the nodule cell. So let's begin to understand the biochemistry, biological nitrogen fixation. after the nodule formation. The end product of biological fixation is ammonia. So the ammonia is the end product of the biological fixation. So the bacteria here and plant, which that is nitrogen fixed into the glutamine and glutamate. So the overall reaction for fixation, for the fixation of molecular nitrogen into ammonia, that is important things. So after this, here is reaction, we can draw here the simple way to understand. The nitrogen in the presence of 8 electron and 16 ATP used to produce the ammonia, two molecule of ammonia and hydrogen. So in this way, the nitrogenase enzyme used for it. And the next step is the conversion of ammonia to nitrate. So that is very important. So ammonia will convert into the nitrate is known as the NO3, not NO2, because you should know about that NO3. So after this, NH3 will be converted into the NO3 and go into the root in plant. So the and absorb a nitrification by bacteria. So this nitrification by bacteria and reduction of nitrate to ammonium is very third step. So inside of the plants, nitrate is assimilated to organic compounds. Prior to assimilation, nitrates is reduced to ammonia in the cytosol. So into the cytosol, it will convert into the ammonia from the nitrates. So the here I am drawing the you can see this is the equation is the nitrate convert in the presence of NADH and 2 electron and 1 proton will convert into the NO2 is the nitrate reductase enzyme to produce the nitrite, and this nitrite and water will be produced. And in this way, the next is the B step is the NO2 is the basically nitrite will convert rapid transported into chloroplast when it is reduced to ammonia by the activity of nitrite reductase. Ferredoxine is the electron donor. So the ferredoxine will donate the electron. Here you can see the nitrite will be 6 ferredoxine reductase reducing to oxidize. So in this way, the oxidize to produce the ammonium ion, and this ammonium will be the corporation of ammonia into organic compound will be formed. So the ammonium will be absorbed, and it is here is the incorporation of ammonia into organic compound is alpha ketoglutarate with the ammonia to convert into the glutamate, and here the glutamate in the presence of ammonia again with the using of ATP and glutamine synthetase enzyme used to produce glutamine, and this glutamine with the presence of alpha ketoglutarate to produce 2-glutamate, and this transamination reaction will be occurring the next. In this way, the glutamate and oxaloacetate will convert into the aspartate and alpha ketoglutarate. So the glutamate aspartate are metabolic reservoir for temporary storage of excess ammonia. So in this way, the ammonia will be assimilated into the plant in the form of glutamate and oxaloacetate. This was the biochemistry about the biological nitrogen fixation. So I hope you make sense about this. Thanks for watching. Subscribe this channel for more updates, and also give the comment below for understanding these things and give the feedback. And if you like this video, please thumbs up and subscribe, share to your fellows, and comment about the new video about the nitrogen cycle details. So thanks for watching. Bye.